REEF has allowed me to build a larger community of marine conservation supporters worldwide by connecting people to the ocean through the Volunteer Fish Survey Project.
I've helped with the mitigation of lionfish through publications, outreach, and removal actions.
I've gained first-hand field experience building deep water lionfish traps and educating the public about invasive species.
Through REEF, I've been able to share my passion for marine science with others by leading education programs both in person and virtual.
REEF has allowed me to spread diversity and inclusion through marine science by highlighting the voices of minorities in the field.
REEF is committed to using our program's platform and voice to build a better world for the conservation community by reducing barriers and supporting accessibility for all. The Oceans for All Fund is a pooled scholarship fund supported by individuals, businesses, and foundations committed to investing in a more equitable future for marine conservation. By partnering with REEF and adding your voice to this crucial work, you are helping the next generation of ocean stewards gain experience.
Through REEF, by kindergarten and first grade students, we're able to learn about the invasive lionfish and their impact on the ecosystem.
Go in the ocean to clean up the invasive species.
Creating inclusive opportunities for others to cultivate connections with the ocean regardless of zip code.
Well, my favorite part was snorkeling.
Coral reefs, my first time.
We got to see a lot of wildlife, animal, and sea creatures, and it was really beautiful.
Together, we are making a difference in the health of our marine environment by increasing access and removing financial barriers through facilitating virtual field trips at no cost, issuing educational scholarships, and providing financial assistance to our marine conservation interns.
Because of my REEF internship, I've been able to build connections and explore new opportunities to advance my career in marine sciences.
After learning about the Nassau Grouper, I want to protect the environment. There is no more overfishing.
You can make a difference by participating in REEF's Oceans for All Fund. You can create opportunities for the next generation of ocean stewards and supporting foundational change in how we work going forward.
REEF is exploration. REEF is empowering.
REEF is impactful.
REEF is preservation.
The ocean's more important than you think.
We love REEF!
Back up them.
Thank you all for coming back for Saturday through the rain. I know the skies opened up kind of right when everybody was probably trying to get out of their cars. So appreciate you being here. We are super honored to welcome Dr. Richard Smith as our speaker for this afternoon. Before we welcome him up, I just wanted to say once again, thank you all for helping us celebrate Reef Fest this year. It's really been a great, fun celebration. You know, that community that Martha talked about yesterday, lots of fun bringing everybody together. If you haven't had a chance, we do have a visitor survey. If you can fill that out, we'd really appreciate it. Trying to kind of do a little bit better job of keeping track of how many people and where folks are coming from. We do have a prize. We're going to draw a prize. So if you haven't had a chance, those are at the welcome desk. You could just, it's a real quick 5-second kind of survey. We want to extend a special thanks to the folks at Dancing Goat Coffee, right? For those of you who have been to this event before, you know that's a new addition and a welcome one, a really great coffee. She generously is donating all of the coffee, all of her time, bringing all that in. So we really appreciate that effort, and hopefully she says she'll be here next year, so yay. And let's see, for those of you who are coming for the Love of the Sea tonight, remember the gates open at about 4:30. The event will officially start at 5, and it's casual. We're going to be outdoors. There's a lot of tents, so even if it sprinkles a little, we should be just fine. Practical shoes, it's grass and gravel and uneven ground, so no high heels. And let's see, I think those were all the announcements that we have. So without further ado, I'm going to welcome up Anna, and she is going to say a few words.
Good afternoon, and before we introduce Richard, I had a message from one of our co-founders, Paul Heumann, and he was very sorry that he couldn't join us this weekend. He's still recovering from a back injury, but He wanted everyone to know how much he appreciates everything you've all done over the years for REEF, how proud he is of this organization, and how much he misses being here this weekend with this wonderful community. So I wanted to share that message with you. And then it's my pleasure to introduce Richard Smith. And Ned, our speaker this afternoon, and Ned and I met Richard. We were talking about it maybe 13 or 14 years ago through a mutual friend. We had been diving with this friend for the first time, and after a few days, she said, "You guys really need to meet my friend Richard. You dive exactly the same, you look at the same things, you get excited about the same things." So we thought, okay, and she made an introduction by email, and Richard lives in the UK. We're back here in Florida, and we emailed, and it's Richard Smith, who is an award-winning photographer, writes a lot of articles for dive magazines, leads a lot of dive travel, and Richard was coming to the US for on business, and we met for coffee, and then the conversation carried over to dinner, and we found out, yes, we all really love the same things. We get excited about the same things, and he's a real naturalist and can tell the stories the way that we all like to hear them. And we were able to go on a trip after that to Indonesia with our mutual friend, and that led to even more exciting adventures, and we ended up going to Japan. Richard organized a trip, and we had a great adventure in Japan, and then we went to, a few years later, to Australia and had even bigger adventures, including like 7 flat tires over a month. So we've been through a lot together with Richard. But Richard is also a special person who's bridged this gap of communication because he also has a formal education in the sciences. He has a degree in biology and a degree in tropical marine ecology and evolution. I got that there. And also his Ph.D. is in pygmy seahorses, the study of pygmy seahorses. So if you've read anything about pygmy seahorses, you've probably seen some of Richard's work. He's also described a couple of, been on the team that described a couple of pygmy seahorses in the past couple of years, including a very exciting one that I'm sure he'll tell you about. He's on the IUCN Specialist Committee for seahorses, pipefishes, and sea dragons. And he's also involved in another citizen science organization. He's a consultant for iSeahorse.org, right? Richard, and because of that formal background and his wonderful love of the natural environment, he's able to really communicate and tell the stories that we all like to hear. So it's my great pleasure to introduce our dear friend, Dr. Richard Smith.
I'm going to break the mold. I'm going up here. I'm so excited to be here at Reef Fest. We've had a couple of near misses with, COVID happened, you may have heard. I couldn't get here last year. There was British people weren't allowed into the US, but finally I've made it. I'm very excited to see the dives around here, and we've had some adventures already. But today I'm going to talk about the wonders of the ocean, the world beneath. The world beneath is the title of my talk, of my book, sorry. But I put this wrasse up here. This is named in honor of, not by me, but the other co-founder aside from Ned, Paul Human. It was named after him. It's so beautiful. And no, he can't be here. He's got a spirit animal at least. Look, I found such cute photos of me. Often people ask me, like Anna said, I'm from the UK. And I grew up in the most landlocked part, right in the middle in the Cotswolds, if anyone's been to that. It's like little cute cottages and things. Nowhere near the ocean. I didn't really have I wasn't near the ocean at all as I grew up, but we had an amazing zoo next to where I live. So there's white rhinos and penguins. So I was there all the time, but also just being out and about and learning about natural history was so important and informative for my life and moving on to where I am now. You got to see cute pictures that I just found myself, so that was good. But I guess when I learned to dive, I was 16. I learned with my dad, and like everyone, I started looking at the big creatures. And over the years, I've been lucky enough to see whale sharks, obviously the biggest fish in the sea. Basking sharks, I was very brave and went snorkeling in Scotland to see those. I don't know, I really should have checked if I never know the conversion between feet and Fahrenheit and Celsius, but it was about 10 degrees Celsius, the water. It was really crazy chilly. I don't know what that is, I'm afraid. And, you know, like the mantas, everyone loves these big charismatic creatures, but they forget then sometimes that the little creatures are also equally charismatic. You can see, oh, I wonder where the zapper is. There's a little ghost pipefish here. There's a lot of little really cryptic creatures that are pretending to be something else. And this is a little juvenile frogfish. So these, as soon as I started seeing these little creatures, and I know it's kind of sacrilege with the fish counters, but I was inspired by nudibranchs, and those really got me looking at the really tiny little creatures, which then ultimately led on to my PhD on the pygmy seahorse biology. I'd done my undergraduate in zoology in Southampton in the UK, so that was on the coast. And then I moved to Australia and did my master's and PhD, and that was on the biology of pygmy seahorses. And I was inspired by the speakers yesterday, and I'd like a show of hands for who has seen a pygmy seahorse. Oh, very good. Good turnout for pygmies. So people have obviously heard a little bit about these guys. They're fairly newly discovered. So the very first species was discovered in 1970, Bargibant's pygmy seahorse. They live on Gorgonian sea fans, so these things can be as large as a car windscreen. And they were discovered in New Caledonia when a researcher collected the Gorgonian and brought it up to the surface to put into the museum. And when they had it, when they'd collected the specimen of the Gorgonian, they noticed on the surface there were these little tiny seahorses. So literally no one had heard about these animals until then. And then over those, so it was sort of in the 90s, divers started to see them in far-flung places like Papua New Guinea. But then since the turn of the millennium, there's been another 7 species. So we didn't really know very much about them until recently, and no one else has studied them really beyond my research. So there's a lot still to learn about pygmy seahorses. And they're very, very tiny. So of the 8 species, the smallest is 1.4 centimeters. The largest is 2.7 centimeters. And I gather that's 1 inch is about 2.7. So they're between ½ an inch and an inch when they're fully stretched out. And they're morphologically unique. So that's what I'm going to explain a bit. I added this slide after I got here because I'm very excited to see a dwarf seahorse. But there's all confusion between pygmy seahorses and dwarf seahorses. It turns out in the Gulf of Mexico here, there's an endemic dwarf seahorse, which is just over an inch long. They're not habitat specific, well, they have habitat preferences, but they don't live in association with Gorgonian corals like the pygmies do. But they're very small, they seem to live on algae, fairly inshore, and I'm very excited to see them because it's really interesting kind of case study in how seahorses over multiple times in evolution have evolved to be really tiny. So on the left is a pair of Bargibant's pygmy seahorses. You can see the male here. This guy's pregnant. So the males hold the eggs, which I'll talk about more in a second, but they become kind of like a balloon when they're like a balloon when they're pregnant, the males. They get really fat. And because they brood the young within the body cavity, whereas the normal seahorses, let's say, have a brood pouch on the tail. So this is a male, this is the opening to his brood pouch. The pygmies, the opening is down here. This is where, once I've got a picture of a dwarf seahorse, I will insert it here. But until then, we've only got the thorny seahorse. So there's kind of this distinction. The pygmies also have other adaptations for being so tiny. So they have a little hole, just one single gill opening on the back of the head for breathing, and normal seahorses have a pair of gills. And there's a few other little quirks, but the pygmies are quite different. So they're different than the dwarfs that you get here. This is the very smallest of the pygmy seahorses. This is Satomi's pygmy seahorse. So far, it's only known from Indonesia, but the more you investigate these little tiny creatures, the more you kind of realize there's so much out there. And this is sort of scale. So compared to a dime, you can almost get two stretching across. So they're pretty incredible creatures, but probably they're further afield. They're probably in the Philippines, I imagine, maybe New Guinea, or Papua New Guinea. But at the moment, we only know them from Raja Ampat and Borneo. But I've seen them only a couple of times, and they come out at night and swim around, and they're pretty incredible creatures. So seahorse reproduction is probably the most well-known thing about their biology. I've been fascinated by this. It's such a weird quirk in nature. They're monogamous, so pairs of seahorses mate at least for each brood monogamously. They don't mate outside of that pairing. So normally if a male mates with a female, neither of them will remate until after that brood has been released. Often the pairings can go on for years and sometimes a lifetime. So that's obviously why it's kind of fascinating. But the other fascinating part is that the males become pregnant. So the females produce unfertilized eggs, and during mating, pass them across. They join up and they pass the unfertilized eggs across to the male when he fertilizes them and as they enter his brood pouch, which is such a weird quirk of nature. And it's basically the reason that the males put so much effort into raising the young is that they know every single baby is theirs, which is almost unheard of in nature. Before genetic techniques, a lot of birds were thought to be monogamous. When it turns out we did genetics, there's been a little outside of things going on, but seahorses never have. In this picture, you can see in the foreground is the female. She's much more slender because she doesn't brood those eggs, and the male is in the background, and you can see he's quite swollen. He actually gave birth the next morning. But these guys, every pygmies at least, they get together every dawn and dusk and do like these little courtship dances. And those are really important because this helps to synchronize their reproductive system. So the male will meet with the female, they'll do the little dance, and he'll pass across the information that he is going to give birth soon, or that his brood pouch will soon be ready, and that she must start preparing some eggs that he can take into his pouch. So she will start to hydrate some eggs, in which time she takes on a bit of water and the eggs swell in size. Her size increases a little bit for 4 or so days, and then he'll give birth and she'll pass over the eggs. But yeah, it's fascinating watching these little guys. So I watched the pygmies every dawn and dusk doing their little behaviors and things. Not all of them, not the bigger seahorses aren't every day, but yeah, it's a fascinating thing. This whole project of my PhD, I'm just going to show you a very tiny bit of it. Obviously, it would go on and on. If I was telling you the whole thing, you'd be all fast asleep. But a fascinating part was the social behavior. So I wanted to investigate the social behavior of pygmy seahorses. And actually the whole project came to me looking at a group of pygmies that were living on a Gorgonian coral. So this is Denise's pygmy seahorse. And I was looking at a group that had 3 individuals. But I was really interested because these guys live on these Gorgonian corals, but I was a little bit confused. So if they have these monogamous pairings, if you have 3, there's obviously a third wheel, and what was going to happen to that one? I was really fascinated to investigate this. So looking at this one in Papua New Guinea, I then started to sort of gather all these questions. So my project was on these 2 species, which is Bargibant's on the left and Denise's on the right. They're both obligately associated with Gorgonian corals. So the one on the left, Bargibant's, only lives on Muricella gorgonians, and that's only in the coral triangle area of sort of Indonesia, Philippines, New Caledonia, as far south as they go. Actually, when we were in Japan, we saw them also near Tokyo, which was a huge range extension for them. About 1,000 miles, I think, further north than they'd been recorded. And Denise's is much more cosmopolitan and relatively generalist. So these guys live on about 10 different genera of Gorgonians and even whip corals. So those of you, I know there's probably not everyone's a diver, but Gorgonian corals, if you're not familiar with them, are these huge, big, flat corals, and the pygmies are perfectly camouflaged for the specific Gorgonian that they're living on. And so they can imagine they're really difficult to find and quite hard to study. But yeah, this is a Gorgonian coral. So I was based in Wakatobi for this study, and it was really important to be able to just have access to a reef that I could access basically at any time of day or night, because I wanted to look at this social behavior, and a lot of it was happening before the sun came up or after, or as the sun was setting. So I couldn't assume that anyone was going to take me out on a boat to wherever to any old whim. So this was perfect. I could jump in, and I found a group which were really interesting for studying my behavior, my social behavior and reproductive behaviors. On the Gorgonian, there were 4 individuals, and can everyone, who can see 4 pygmies? Okay, good. You passed the test, the 4 of you. But there's one here, 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 and the 4th one's down here. So the biggest of these was 2 centimeters, so like 3/4 of an inch. But they spend their whole adult lives on a single Gorgonian coral, so it's very easy to then keep going back to the same place and spend a lot of time studying them. So the friend that introduced Ned and Anna and I was called Wendy, and I met her when I was doing this, actually just before I started doing this work. And I would come back, and I'm a proper scientist, you know, so I would come back and I'd be like, oh, individual 1 was doing this to individual 2, and I had numbers for all of the pygmies, but Wendy just started to get really livid one day when I was telling her this outlandish story, and she said, no, they can't be 1, 2, 3, and 4, they must be Tom, Dick, Harry, and Josephine. So those names stuck. And ironically, it's strange, I'm from this tiny, tiny village in the Cotswolds in England, and Tom, Dick, and Harry were killed in my village, which I only just found out during lockdown, but they were like vagabonds and they got hung for stealing a sheep, I don't know. But yeah, anyway, that was a funny story. So I had Tom, Dick, and Harry, and Josephine. They were 3 males and 1 female. So this was really, really an interesting group to study what their reproductive style was going to be and how Josephine was going to pick her knight in shining armor, let's say. But it was really important that I was able to tell then who was mating with who. And for Josephine, this is Josephine here, you could always tell when she was preparing to mate because she had that egg hydration that I mentioned, and she would start to swell in the belly. She was about 1.6 centimeters long, so it was small, but this is where having my photography really helped. But yeah, she would really start to swell, and it was much more subtle than the males, but after she transferred the eggs, she was certainly much more slender. Although with the males, it's very easy to tell when they've given birth. So this is Tom, and Tom just became like a football, like really swollen, full of babies, as his pregnancy went forward. And inside the brood pouch, the male really is pregnant. So there's all sorts of blood vessels and things within the brood pouch, and he's bringing nutrients and oxygen and removing waste and things as the babies develop in there. But I think probably he's the only male in the animal kingdom that gets stretch marks after he's given birth. So for a few days, and even after I'd found individual males on their own on Gorgonians in a different part of my study, if they'd ever been really pregnant, you could always tell that they had been pregnant in the past. I was really lucky enough, well, it took hundreds of hours recording every behavior, but to figure out exactly when they were going to be giving birth. So this is Tom early one morning releasing his brood. So this is 2 of them, and you can see they're zoomed in here. The little shiny bit is one of the baby's eyes, and there's 2 little babies, and the tails actually wrap around the baby's snout. But you can see the babies are really dark in color, and this is because when they're born, they then float away in ocean currents. They don't stay on this same Gorgonian. They spend 2 or 3 weeks floating around in ocean currents, and like Ben was saying yesterday with the anemone fish, it's likely that they then use chemical cues to find, again, new Gorgonians to settle on. So yeah, that's why they're dark in color. And really, there's no rest for the wicked with these guys. As soon as Tom gave birth, he literally swam right back to Josephine, who you can really see is absolutely full of eggs. In this picture, she's full of those eggs that she'd hydrated. She was really, really full. And Tom had just given birth, so he'd pumped his brood pouch full of water to flush it through, but that's all. And then 20 minutes later, they were mating. And you can see her size reduces as the eggs go in, and his wrinkly belly becomes full of eggs again. So it would be exhausting to be a male pygmy seahorse. But in addition to this, I was looking at other behaviors, obviously. I saw something called neck wrestling between them. Well, I decided it was called neck wrestling. That's what's happening here. The males would try and knock each other over with their necks, because they obviously don't have fists to punch each other. So they were trying to size each other up with their necks. They also strangled each other. So here, this is Tom and Dick strangling each other. This is Harry. So it turns out that you can just about tell in this picture, but actually both Tom and Dick were pregnant. So it turns out that Josephine was mating with one of them on day one. The pregnancy would last 2 weeks, just under. So that would be 14 days that Tom was pregnant. Then 7 days through that, she would mate with Dick, and basically alternating. She was constantly mating with the two males. They were constantly pregnant and constantly producing young. But Harry just was kind of smaller than the others. You could really tell he was smaller. And he just sort of sometimes watched as those other males had these crazy encounters and was maybe learning for when his time came. But yeah, he never did mate in the time that I was studying them. And they did have a huge epic tussle one day. So this is Tom, Dick, and Harry. So they were all fighting, and this one lasted about 5 minutes of thrashing around as the three males were fighting it all out. So this is just one of the groups that I studied, but it was just amazing. So after I finished my PhD, I was honored to be invited to join the IUCN Seahorse, Pipefish, and Sea Dragon Specialist Group. The IUCN is the International Union for the Conservation of Nature, and it's a non-governmental group that we help do like red list assessment. So, you know, if an animal is considered endangered or something, we do that for the seahorses and pipefishes. And if there's ever any sort of international issues with seahorse conservation or anything across the world, we get involved with that. So it's a huge, there's about 30 of us that are the sort of experts in the seahorse world. And they're from all over the world. It's such an inspiring group of people. But once I was invited to join that, it sort of opened my world beyond tiny pygmy seahorses. And I'd seen this little Japanese pygmy seahorse after I'd been to a conference in Okinawa one time. I went to this conference, got the funding to go, and then legged it up to Tokyo because I'd seen these pictures of this seahorse. I knew that some Japanese people had been seeing this species, but no one had really seen it much for about 10 years. And I had kind of a hunch that it was different. So I managed to find about a dozen of them on this little island called Hachijo, which is about 200 miles south of Tokyo, quite cold water. But eventually there was about 5 continents worth of researchers. We got together, a Japanese guy managed to collect for us, and he was on our group that then went on to name this seahorse in about 2018. So yeah, it's just an incredible little guy. And although they look so similar to the other seahorses, these guys don't live on Gorgonian corals. They live on sort of algae and things. But they split about 7 or 8 million years ago from the other pygmy seahorses. So although they look very similar really to the others, they are indeed quite different. This picture is actually, I was with Ned and Anna. We went on an adventure to Japan to look for these guys. And I was like, oh yeah, I saw a dozen last time we were there. It'll be easy. That was stupid, wasn't it? There was all sorts of hurricanes. We'd planned to be in Hachijo for a few days, or a week actually, but then there were hurricanes heading our way. We managed to do a couple of days of diving. I think it was probably the third or fourth day. We still hadn't found the pygmy seahorse, and our guide, Kotaro, finally spotted one on the last 10 minutes of the last dive before we had to leave, and that was our thing. And he was just suddenly like so elated on the dive to spot this guy. So I was glad that we managed to share this cool pygmy seahorse. And through the specialist group, I've also been able to help out guys working on this New Zealand pygmy pipe horse, which is another new species just named last year or this year, I think. Incredible little guy. And we're working on, this one's from South Africa, another pygmy pipe horse that is not yet described, but we're working on that probably next year. That'll be published. So after my pygmy seahorse PhD, I was really like inspired to be looking at where animals are living. And I sort of was learning how specific everything is, not just pygmy seahorses, but on these coral reefs, every niche is filled. And that's what's creating that huge biodiversity on the reef. So I was, for instance, in this type of leather coral, I'd been looking for this type of pipefish for years. In one of the old pipefish ID books, there was a little pipefish that was banded and sort of beige in color, and it was the only picture I'd really ever seen of it, but it was on sand, so I knew that wasn't the habitat. But it's so fascinating that as soon as I figured out that these guys live in this leather coral, I actually found them in almost every one of those leather corals, and they were just there all along, and you just kind of have to know the habitat. And that once you've got that key, I mean, I'm sure for the reef surveyors, you guys have your specific places. You're looking in little crevices for a certain type of blenny, or you know which habitat to look for certain things. But this was a real like eureka moment. But you must look for these guys if you're ever in, these guys are in the Indo-Pacific in the Coral Triangle, but if you're looking in there, look for their little silvery eyes. You don't need to use a light, but you see the shining little eyes chipping about in those corals. Another one that I'd also looked for years and had never figured out the right habitat. And then I forget actually who it was, but between like the little group of us that got so excited about these things, someone figured out that this is the type of coral that another pipefish lives in. And again, so many of them are inhabited with these pipefishes. Those coral polyps are about the size of my little fingernail, and the coral colonies are about the size of my fist. And amongst those little coral polyps, there's a pipefish. Actually, there's 3 species of pipefishes, each specific to a type of similarly related coral. And they just go between them, and they're just these incredible little guys, but they're, yeah, again, you just have to know exactly where you need to look for them. I'm going to embarrass my dear friend Janet Eyre, who some of you may know as one of the great surveyors of the Indo-Pacific. She and I dive a lot together, and we sort of bounce off each other looking for different things. But she has this motto, no spine, no time. So she's counting fish, she wants to see fish, and if you see anything that's not a fish, then it's just basically dead to her. But we were diving in Raja Ampat in West Papua, and there's a tiny little anemone that called a leopard anemone that you see sometimes. It's not very common. It's about the size of the first joint of my thumb. So it's a small little anemone. But in '99, I think, this shrimp was described from the surface of those little anemones. We were at 30 meters, like 100 feet, and I suddenly spotted this shrimp, which you see here. This is the female leopard anemone shrimp. I just said how big the anemone is. The shrimp is tiny. And I was so excited. I called Janet over. I'm like, Janet, look at this. And she was like, oh, yeah. I don't know if she actually even saw it. She just was like, it's not a fish. I'm not interested. But actually, there's even a little male here hiding up in the top. I don't know if anyone spotted that. But again, it just shows. And these guys, those leopard anemones are really variable in color. So the shrimps also are really variable, like the pygmies, depending on the habitat, they change color to suit it. So I went down a bit of a rabbit hole hunting for crazy things in crazy places. This was a shot that I won the habitat category of Australian Geographic Nature Photographer of the Year. I think it's just such a colorful, engaging habitat. But I took a wider shot to sort of show where they're living. It's a little amphipod that he lives at the mouth there. I didn't know anything about this, so I sent off, I did some research and found out a guy who worked on these little amphipods, and he said that he'd recently described from this area an amphipod and named it after Elton John, which seemed marvelous. I don't know quite why. Apparently they listen to Elton John a lot in their lab. But he explained the biology of these guys. You can maybe see that he's almost like a little praying mantis. He's got quite large pincers, and he's doing that to protect, according to this researcher, inside the tunicate, which is only about that big. There's lots of females and some of his offspring. So he sits at the mouth of the tunicate to stop any predators, like dottybacks often go inside tunicates and things. So he's serving a good purpose sitting there at the mouth of the tunicate. I was just chatting earlier with Anna about this one. To me, this is one of the most incredible animals that just kind of blows my mind about this interconnectivity and habitat specialization. So this is a nocturnal hermit crab. So if you're doing a night dive in most parts of the world, you see these guys, they come out, or actually a day dive too, but they use the old shells of snails and things. The snails died, they then take over the shell. And on the surface of, in this case, the shell, there are these certain anemones that the hermit crab grows. And on the surface of those anemones, Anna found that there's a little tiny porcelain crab that lives only on these anemones that only are found on the shell that only this nighttime crab has. And it just kind of blows my mind, these different levels of biodiversity, really. So being able to take photos has actually really been vital during my work, both when I was studying the pygmy seahorses, and it's kind of helped inform some of the shots that I take. So I'm not really one for actually really the technical side of photography. And I'd seen people trying the biofluorescence photography. I don't know if anyone's ever tried it, but you can cover your strobes and your mask, and you can put a filter on your camera, and then use a special light to shine over the reef at night. And then certain animals biofluoresce, they kind of absorb the light and reflect it in a slightly different way. So during the day, a scorpion fish is almost impossible to see unless you're really paying attention. But when you're doing this technique, they, as you can see in this picture, they absolutely just glow. And it's really strange, the animals that do glow. Some absolutely don't glow at all. And some of them glow in different colors, like the stargazer, I discovered has this eerie green glow. But the biofluorescence occurs across different species, unrelated species. Often it's kind of camouflage species, and it may be that they see in different ways that we don't, and that's what causes it, but we don't quite know why. Oh, can you see that very well? I can't from here. But so certain species do biofluoresce, and they're very, very vivid when you're there looking at them at night. It's really discombobulating, actually. You have all this filters on, you can barely tell what's going on, you couldn't tell which way's up. But you could see from several meters away, for instance, this little triple fin. So if you were wanting to survey triple fins, this would be a really, really good way of doing it, because triple fins are very cryptic and small normally. They're just maybe 1.5 inches. But if you had these filters and things, you could be several meters away and easily count all of them because they're so bright. But someone had studied this and suggested that pygmy seahorses might also biofluoresce in the same way. And another part of my PhD research had been to do population estimates of pygmy seahorses. And doing transects, which is very laborious. I surveyed an area the size of a soccer field, which took a long time. And I counted how many pygmy seahorses there were, but they suggested that pygmy seahorses also biofluoresce in this way. So I was really keen to try this out, because you could look at a gorgonian and within seconds count how many pygmy seahorses there were, which would be so valuable. Turns out that's a pygmy seahorse in the middle of the shot. So that's the pygmy. There's a tiny bit of biofluorescence on the tail. There's like that much on this other one. So it turns out that they don't biofluoresce, and I was devastated. But I think probably, obviously they had seen it, but I think in some cases, pygmy seahorses and some other fish kind of molt their surface, like the skin surface comes off every now and then to get rid of parasites. And I think maybe some algae had grown on the surface of the ones that they were looking at, and it was actually the algae that was biofluorescing. So sadly, that's a terrible technique for estimating pygmy seahorse numbers. But we've found kind of the last couple of decades, last 20 years or so, we've known, historically that these leafy sea dragons have been only found in southern Australia. And we know that there's endemic species that are restricted in their range and where you can find them. But it turns out in the Coral Triangle area, there's a lot of micro-endemics. So that's why, like, Ned and Anna and Paul's books have gone from small and like over the years have grown in size, because there's so many species that we're finding that are really restricted in range. Some of them are quite incredible, but it really does depend where exactly in the world you are. So this is the Coral Triangle area. So this is the world's most biodiverse marine habitats and the most species in the world. Any distance from here, there are fewer species. So it's a really important area. But we found, for instance, this is Raja Ampat. You may hear people speak about that in West Papua. This is a really biodiverse area, and it has many species that are only found in Raja Ampat. This is Chendawassi Bay, a very remote, obviously, area, but one that I've been lucky enough to visit a few times. And researchers only went there for the first time in about 2006, and they found a whole suite of species that are very common there but found nowhere else on Earth. And there's all these different areas around that have all these specific species. A really cool one is the Bangai cardinalfish, and it offers a really good example of how these species are evolving. So the Bangai cardinalfish is naturally only found in the Bangai Islands of Indonesia, which is a very small area, like smaller than Florida, probably half the size of Florida. It's a very small area. But they have actually been introduced into Bali and Lembeh Strait and Ambon, if you happen to have seen them. I imagine no one in this room has seen them in Bangai, but they are known from these few other places. But like cardinal fishes, it's the male that, they're paternal mouth breeders. So the male, once the eggs are fertilized, he takes them into his mouth and carries them round until they hatch. Most cardinal fishes keep the fry in the mouth for just a few days, and then they release them, they spit them out, and they float around in the ocean currents to settle on a new reef. The Bangai cardinalfish, which is just probably only this size, is a really interesting case, because when the fry of the Bangai cardinalfish hatch, the male keeps them in his mouth for 2 weeks. And this was an example where the male had a bunch of little babies. This is one baby, this is another, this is the eye of one of them. It was so cute watching these little guys, as all the babies were desperate to see what I was doing, taking the photo, and they were like fighting their way to the front to have a look to see what this big weird creature was doing. But it was incredible. But so these babies have been 2 weeks in the male's mouth. He then spits them out, but they're far too big and conspicuous to then float around in ocean currents. They'd be picked off almost immediately by predators. So they settle straight to the reef floor. Oh God, I'm terrible at this. Don't forget what you just saw. So they're big and conspicuous, and they go straight to urchins where they're protected and there's spikes, and they then grow, but it means that they can only cross areas of suitable habitat. So the Banggai Islands are shallow, and then it's very deep surrounding them. So they've evolved basically in isolation, and they can't travel beyond that. So that's why these guys are found only in a small area. Recently, the last big trip I did to Indonesia in 2019, 3 years ago, actually, almost to the day, we went to a strange far eastern part of Indonesia called the Forgotten Islands, where we found this new species, which is yet to be described, of dottyback. These guys are mimicking a type of damselfish that looks almost identical with this white tail, dark body. And so this is another one that's only found on a few islands. We actually went to a nearby island where there was another species that was similar, but obviously a different, distinct species. So we know there's new species out there to be named. We also went to Halmahera fairly recently, which is just west of Raja Ampat. Halmahera is a kind of strange island because it's got birds of paradise, which you associate with New Guinea. It's got these walking sharks, which also are only usually found around New Guinea and Australia. But it turns out that one fragment of the landmass of Halmahera had drifted from, in a long geological time, had drifted and crashed into these other islands and carried with it birds of paradise and this Halmahera walking shark. So it's less than a decade ago it was described, but it's a huge fish. You sort of imagine that we know so much already that there wouldn't be sharks that hadn't yet been described. We went to Japan and we saw these amazing Yuzen wrought iron butterfly fish, also only found in the cool temperate waters of a few islands in Japan. And this is a little Galapagos pike blenny. As I said early on, I had those pictures, the whale sharks and things. Well, in this dive was the sea lion dive. You're supposed to look at sea lions, but I was like, had my head looking in little pike blennies, and they were just incredible. They're just the size of my little finger poking up, and the males are displaying to each other. And I was trying to deal with the ridiculous sediment that the sea lions were stirring up whilst they were livid that I wasn't paying attention to them. In the Red Sea, I know there's been a group of you have been to the Red Sea recently, and you probably saw these beautiful Red Sea anthias that they're off on the reefs there. Also, I was recently in the Red Sea. It was a good pandemic spot for the Brits because it's very easy to get to for us. I was there and I saw this grouper that I had never seen in any picture, in any book or anything. It turns out that it's a very regionalized juvenile color form of the slender grouper. So the slender grouper in most of the Indo-Pacific pretends to be, as a juvenile, it mimics this other type of wrasse. In the Red Sea, this wrasse isn't there, but this grouper is. So in the Red Sea, occasionally it seems to mimic a type of endemic cleaner wrasse. So normally they're sort of beige in color, but they're called a cobalt grouper. They've only been seen a few times, but I was so shocked to see such a wonderful animal on the reefs there. I also love an underwater soap opera. I'm fascinated by the incredible behaviors that you can see if you do your dives a little bit slower than you might imagine would be kind of your normal speed, and get a bit closer, really watch the animals and the behaviors that you're seeing. Usually I would really recommend not getting involved in those behaviors, but this sea lion in the Sea of Cortez really was desperate to grab onto my leg, and she just kept swimming down, grabbing onto my leg, having a look right in my eyes, and then swimming off. On the next dive, I actually decided to shoot some of the endemic triple fins that are in the Sea of Cortez, and she wasn't so happy about that, and she put my whole head in her mouth. The first thing I knew about it was the teeth marks on my forehead. She didn't mean malice, she just was like, hello, we've got to play, remember? But the behaviors that I've become fascinated, and there's a little gaggle of us have become quite fascinated recently, is the fairy and flasher wrasses. These guys, around dusk every day, probably 3 or 4 o'clock, as the light gets a bit lower, on certain rubbly habitats, usually a little bit deeper, maybe 20 feet or so, the males start to display, and there'll be 1 or 2 males per small harem of females, maybe 6 to 8 females. The females are kind of pale pink in color, a few inches long, and they'll be much closer to the bottom. But there's a few males that are like the peacocks of the undersea world. This was the flame wrasse that Janet found for me in Hawaii that I'd never seen. It's only found in Hawaii. It's a really beautiful one. But we're finding actually that in a lot of areas, there's endemic flasher and fairy wrasses. So I was on a dive with Ned and Anna, our friend Jan, in Indonesia, in a very strange bit in Alor. We found this little weird cut through, so we just decided to go exploring. And then all of a sudden, Anna was like, "Oh, quick, 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 you must see this." And there was a few males of this incredible little flasher wrasse. There's probably only 2 inches long, but wow, are they incredible. They suddenly at 3 or 4 o'clock, those hormones get going and they just start swimming up and around. Their colors go from pale pink, which the rest of the day they are, and then these colors flash on and they look nothing like this for 22 hours of the day. They're really incredible little fish. It turns out this one was a new species, and a few years ago it was named after our good friend Jan Alfian, so the scientific name is in honor of him, which is lovely. And it even turns out that only a few years before that, in Komodo Island, another species had been discovered and also just named as well, Rennie's flasher wrasse, which is similar but different. And each of these islands seems to have their own different species, and there's still so much more to discover out there. But during this crazy frenzy that happens, there was a term we decided amongst us, that there was wrasse-demonium. When the wrasses, there's so many species, when the habitat's perfect, you often will get various species all mixed together. And during this frenzy, the males get so colorful, the females don't know what's going on. Everyone's like flashing and God knows what. And sometimes the female will pick the wrong male to mate with. And actually you see hybrids very often in these wrasse-demoniums. So this male here, I caught displaying, I spotted him as being quite different from the others. So I'm pretty sure that it's a mix of this species and this species. He's really quite different, but they're all so closely related. I imagine he probably would be fertile, although hybrids often aren't. I think these species are so closely related that he may well be. And for me, I find underwater that there's so much unexpected beauty. I mean, in the UK, everyone hates slugs. They come and eat your garden, and if you've got hostas, I don't know if you even have hostas here, but they eat everything. And I love a sea slug, but they're so beautiful. And I'm going to introduce you to some other things that I think are also terribly beautiful, and I hope I can convince you. Even worms, and I mean, flatworms are beautiful. You tell someone you love a worm and they think you're mad. But I'm going to try and share with you my love of parasites. I hope no one had lunch very recently. I think we're doing okay, I think, time-wise. Hopefully it's digested a bit. But these isopod parasites, like many parasites, are very, very specific, usually to the species, about what they will infect or inhabit. So these isopods, there's many species. In this case, only in Halmahera had I ever seen on this dotty back these types of parasites. The larger one is the female, the little one is the male. And the females, a bit like a kangaroo, they have a little pouch that when she lays the eggs and the male fertilizes them, they go in that little marsupium. They then develop, and then when the time comes, they're released, and they often infect almost all the fishes in a local area. They have like a broadcast boom spawning. So a lot of them, like I'd never seen it before, but almost every one of these dottybacks in this area had them. Often parasites will sterilize their host because these parasites are drinking the blood or feeding off mucus or something. But if you're wasting energy and producing eggs and things, then that's no good for the parasites. So they'll often sterilize the host that they're living on so that they can't produce any energy and that the parasite can then be amazing and everywhere. But so many of them are undescribed. I kind of started to reach out to some researchers. Basically, none of the ones I was sending them had been described, but without collecting specimens, basically they couldn't then go on to describe them, and I wasn't going to carry suitcases full of dead parasites around. But I loved this one. This in Raja Ampat, I'd never seen it anywhere else. This fusilier has a little parasite that lives underneath the chin. What I also found fascinating was that it sort of has this counter shading. So normally, almost always, they're black in color and they sit on the surface. But this one seemed to know that, or through evolution at least, it's evolved to become white in the similar sort of colors as the fusilier, so that the parasite's host then isn't going to get eaten. If the parasite was black in color and lived on the white fish, predators would spot that host and eat it. So natural selection has meant that this little parasite has also become white. I also love this picture because I love the face of the little cleaner wrasse, because he's like, oh no, I've missed the ball, like dropped the ball here. Obviously this parasite's way too big for him to be any use. They have to catch them when they're really, really tiny. Another cool parasite that I saw was living inside the nostril of a crocodile fish. I've only ever seen this in one place in the Philippines, and I spotted this crocodile fish. They get to like 3 feet long, and one of them had a massive nostril all swollen. And when I looked closely, I could see that this is the tail of one of those isopods. So it seems like a marvelous place to go. You're very safe. You can drink some blood. So yeah, that was another one, also on new species. Probably the most famous and ew-worthy one is this tongue biter parasite. This is the parasite living in the mouth of an anemone fish. As when they're very small, these parasites climb in through the gills of the fish. They then go into the place of the tongue and kind of, it's not quite known if they eat it away or kind of kill it off, but they bite on then and make their place. The male will, that's the female, the male will be further up the throat of that fish. And it's apparently the only artificial prosthesis in nature, because the parasite kind of helps move the food around, apparently, with the tongue as well. So it's serving a nice purpose too. There's not too many more parasites if you're feeling queasy, I'm sorry. But this is also another incredible one. I took this picture and I kind of had a feeling I knew what it was, but I was in touch with the guy from the Natural History Museum in London, and sent this picture, and they apparently had only seen this in a crab in the British Channel before. And this was taken in deepest, darkest Indonesia. This is a fecampid worm, and it's a parasitoid. So this worm infects the shrimp, and the pink stuff actually, yeah, is a worm. It's not its intestines or anything. But what the parasite does is slowly, starting with the least important part of the organs or whatever of the shrimp, it eats that, then it eats the next least important, until it's basically eaten everything that it can, that whilst the shrimp can still keep functioning. It basically takes up the whole of the shrimp internally and then bursts forth some eggs and infects a whole new generation of shrimps. So it's good that we don't have those sorts of things, I think. Another kind of shrimp that parasite that I'd been fascinated for years is this little lump that caused this, well, the parasite that caused this lump underneath the shell of these little shrimps in Indonesia. I'd seen this lump on so many different ones, and I was really, really fascinated to investigate what exactly it was going on under there. Finally, I realized that there's another shrimp that does get infected with this isopod, which is here. So this shrimp has a transparent carapace, and I could then see what was going on inside. And over several years, every time I saw these parasites, I would take a shot, and I could then see, so instead of having the marsupium where they lay the eggs inside the pouch in the body, these are laying against the outside of the shell. So you can see all of the eggs of this female there. On another occasion, I found these little baby parasites. So this is the mother. So those babies had kind of developed a little bit more. And you can see here 4 little juvenile parasites as they're developing too. And this is the final parasite I'll stress you out with. This was a copepod that we saw in Japan. This bean is the parasite, and it seems, because they're so specific, that it was actually biting onto the eye of the scorpion fish, and that's probably where they've evolved to bite onto. They'll only bite onto the eye of certain, probably only this one species. This coil is the egg coil of that parasite. But what I love is that there's a little barnacle. I only saw it when I zoomed in on the photo, but there's a barnacle which probably only lives on this type of parasite, which only bites onto the eye of this type of scorpion fish in Japan. I just find that incredible. And yeah, there's so many incredible animals and organisms that I've seen and we all see over the years. But I kind of just want to finish with where we're going with all this. I was lucky enough to dive just before the first coral bleaching event in the Maldives in '98, when it was much like the previous photo. I then went back there in sort of 2014 time, and many of the reefs didn't recover from that terrible coral bleaching. Some have, it depends on the local conditions. But in the Indian Ocean, in that very first coral bleaching event in '98, some areas had 90% mortality of corals, which doesn't recover in 20 or so years, as it turns out. Coral bleaching, for those of you who've not heard of it, occurs on hard corals. So they're comprised of many individual polyps, and within the cells of those polyps are zooxanthellae, which are a type of little algae. Those algae use the sunlight to generate energy and help those corals grow in that really nutrient-poor area. So they grow on, and it's that symbiosis that sort of fueled this growth of coral reefs. And coral reefs obviously have huge diversity of species. The corals kind of produce a diverse structure for animals to live in. So not all the species rely directly on living corals, but they do need the structure to hide in and things like that. Many species are really, really specific about where they live, as I've spoken about. Butterfly fishes, for example, have really modified mouths to feed on coral polyps. So they're a good indicator. And for example, reefs work would help show where butterfly fish populations have fluctuated, which specific species are present. And that gives you an indication then of the corals and how they're doing. In Japan, there were huge bleaching of the fields of Acropora corals. These guys are really specific about feeding on coral polyps, so they disappeared within just a couple of weeks of that coral bleaching. And there are other things like little hermit crabs need healthy corals to live. Even anemones, like Ben was speaking about yesterday, they also can bleach. They seem to be kind of a little bit of an indicator. They're often early on. If there's a coral bleaching coming, they can be some of the early ones that bleach. But they don't always die. That's the good thing. Sometimes bleaching a coral or an anemone can go white like this, and that's where the term bleaching comes from. It's when they become very pure white. But sometimes new algae do go in and reinfect them, and things are fine. But even if an anemone fish lives in a bleached anemone, they have reduced output of juveniles. They don't breed as much. They don't have as many healthy babies and things. And even although gorgonians don't have those zooxanthellae, warmer water temperatures can kill off gorgonians. So in the Caribbean, one species of gorgonian, when the waters got warmer, there was a type of pathogen that did well under those conditions and wiped out almost all of that species of gorgonian, which would be horrendous for Bargibant's pygmy seahorse, for example, that relies exclusively on that one species. This was Josephine. Part of her gorgonian had a little dead bit on, and she insisted on landing on this bit of dead gorgonian, and then would get stuck to it, and then would thrash about, and I was holding my breath. Don't get eaten, you're so important for my study. Of course I loved her. Obviously also lots of pollution in the oceans. This was Cendawassi Bay, you may remember from that picture, the very remote place in northern Papua. But you see often diapers and plastic bags and crown of thorns starfish which eat hard corals. And this one also was particularly poignant for me. This is a new species of damselfish, the Chendawassi damselfish, was described, I think, 2012, only known from Chendawassi Bay. And I was kind of just a little bit heartbroken to see it. These are all its eggs, and it's rearing its brood on a plastic bottle. You just don't want to see that in these beautiful, far-flung places. But of course, there is hope, and that's what we must look towards, is the ocean continues to surprise us. And my biggest surprise, probably to date, actually, is seeing this pygmy seahorse. Some researchers were in South Africa, and the pygmies that I'd always studied had been in the Indo-Pacific, in that Coral Triangle area. So they're Indonesia, Philippines, New Guinea. No pygmy seahorse had ever been seen anywhere in the Indian Ocean. So when my friends sent me a picture of what someone thought was a pipe horse, they were like, we've got a suspicion what this is. Can you confirm it? And it turns out to be this guy, which is a South African pygmy seahorse, which is just incredible. It's like, I can compare it to finding a kangaroo in Norway. It's just so not what you would expect to find. So this is another one that's just under an inch long. It lives on the really huge rolling, crashing waves, huge surge. And there it is, just the female of the pair that I saw was at one point buried up to her neck in sand because it was such a horrific, roughy-tufty place to dive. And here, this is a little juvenile that I found, really, really tiny, tiny baby. You can see the grains of sand. But so it's good then they're breeding, and we described this species in the pandemic, the first year of the pandemic. We finally got the name out as Hippocampus nalu. So again, I work with colleagues from South Africa and across the world, actually. So yeah, I hope you've enjoyed learning some of the things that I find so fascinating. If this is interesting to you, this is what I sort of wrote about in my book. But yeah, I hope you've enjoyed it. And I believe we'll have questions. Does anyone have a question? Surely someone wants to know how long a pygmy seahorse lives for. I'm sorry if that's your question.
Hello, I was just wondering what resources you use to find like habitat for the small critters that live in them.
Yeah, it's a lot of trial and error. You're looking in different habitats all the time. You kind of do get an inkling for where you're going to find something. If it's a big, like the leather corals and things, you kind of get a feeling that there must be something trying to exploit that habitat. And that's what you do find a lot in the Indo-Pacific, is every sort of niche is exploited. I don't know where we're going to find the next pygmy seahorse, though, having said that.
The South African one, which part of South Africa did you find it?
Oh, I feel like you have a vested interest in that question. It was in Sodwana, so up on the northeast coast near the Mozambique border. So it's kind of not fully tropical. There was sort of reefy patches. It's an interesting place to dive. Usually people are looking for the bigger things like sharks and stuff, but there's lots of cool critters there too.
Yeah, I was wondering, because that's subtropical to tropical up there.
Yeah, it's
not that difficult from the triangle.
Yeah, it's kind of right at the edge, but also the Japanese pygmy that we described a couple of years ago is also from that kind of edge of it's sort of subtropical. So yeah. Good question. I don't know. I think about a year, but yeah, thank you. That's what I can say.
So I wanted to know what your method for being able to see these. Are you looking through your camera lens? And if you are, how many magnifying how much magnification do you
I feel like during lockdown, my eyes have got worse. I've gone now into getting towards my mid-40s now, so I'm nervous about my eyesight. But no, I'm just looking through my mask, just normally, and then looking fairly close. But I think I was talking with someone the other day about search image. I think your brain starts knowing what to look for before you even know. So I'm seeing things with my brain search image before my eyes have really seen it. And you can see even things, like I could spot a pygmy seahorse on that chair there because my brain just knows without really looking.
It's kind of two questions. I'm really looking forward to reading this, but what is your next project? And the second part is, how long did it take you to compile and write your book?
So the compiling took, there's a couple of pictures in there actually that I took on slide film, so a while. There's probably people in here who don't know what slide film is, which is really upsetting. But yeah, like in there, there's a picture of a flounder eating a blue ring octopus. So I could take the rest of my life. I don't think I'd see that happen again. So I'm like, okay, you have to put the slide picture in. So that's, I think, probably almost 20 years of photos and experiences. The writing itself, I'd been kind of plotting the book in my head, and then the publisher kind of went, you've got 5 months, but I was away in New Guinea for 3 of them. So it took about 2 months in a blind panic to write. Well, what was your other question, sorry? Oh, the next project is describing the South African pygmy pipe horse. I put a slide in there. It's kind of a dark brown chap. So yeah, we're working on naming that.
Are there any pygmy? I know we have dwarfs here in the Gulf of Mexico. Are there any pygmies in the Caribbean at all?
Well, I said there were none in the Indian Ocean to people who asked, so I'm kind of nervous to say definitively, but I don't think so, no. Yeah, there's the dwarf is the nearest. There's another little cool dwarf in the Red Sea that I've been looking for years, so I'm hoping we'll be more successful in the next couple of days looking for the dwarf here.
Well, Josephine was a female and Harry was smaller. How did you differentiate between Tom and Dick?
Oh, well, yeah, it's a good question. Actually, they're really different when you are spending like hours a day with them. The pygmies, they're dark in color, like I had in the slide, when they're born, and then they spend a couple of weeks. And then I found one that had settled to a Gorgonian coral. And it just took 5 days to go from black to the exact color of the Gorgonian. And they vary their surface texture as well. So if the Gorgonian coral, the polyps are when they're closed, the surface is smooth, the pygmy will be smooth. But if the polyps are sticking out from underneath, then the pygmy grows lumps all over its body. But it's not an exact science. So there is variation between the individuals. And Tom and Dick one was slightly bumpier and a slightly different color, which when you've spent so long with them. But I also didn't mention in this talk, but it was quite difficult also to know the gender of them, to know whether they were male or female. It turns out you have to take a really, really close-up picture under their belly to see the males that have a little slit where the babies come from when they're born, and the females have a little raised paw. So yeah, it's quite involved.
The pygmies are so tiny. I imagine they're not very good swimmers. Do they spend their whole life on one Gorgonian, or do they move from one to another?
They actually do spend their whole life on one Gorgonian. Gorgonians can be quite far apart, and pygmies are preposterously bright colored. So if they did move between different ones, I'm pretty sure they would be spotted and eaten. So yeah, they do stay on just the one. But they actually are fairly strong swimmers. Like when Tom was off to give birth in the morning, they like to do it a lot of creatures give birth or spawn when the currents are at their strongest, because those babies then get swept away from the reef and to populate new areas. So they often pick a currenty time. And I held my breath watching Tom swimming off to the edge of the Gorgonian to give birth. It's like, please don't get swept away. But he was fine with it.
And you mentioned they spend their whole life on one Gorgonian. How long is that life?
I'm going to say a year. I'll just say it.
So you mentioned that they were monogamous, but then the female was mating with multiple males. Is that pretty common?
No, so this was the first time actually it's been recorded, and the only time as far as I know. But the pygmies are quite different. So eventually between us, I think we might be splitting them off to keep that monogamy within the genus Hippocampus, lest everyone's head explodes. But yeah, the pygmy seahorses, yeah, in that case at least there was polyandry.
Are you possibly motivated by looking for something that hasn't been discovered yet? Is that partly why you do what you do?
Absolutely, yeah. I'm constantly looking for the next different thing, or where could we find it? I mean, there really is so much more out there. It's just incredible, particularly the Coral Triangle, but obviously beyond to South Africa and Japan. Like the Japanese pygmy was within spitting distance of the world's biggest metropolis, and it was a new species. So yeah, it's incredible out there.
What distinguishes a seahorse from a pipe horse?
It's
sea pipe.
Yeah, kind of morphological stuff. Very specific coronet shapes, how they hold their head. But actually, the New Zealand one, you may remember I put up there, it was in a new genus, but actually someone had described a new seahorse from the southern Indian Ocean about 4 or 5 years ago and described it as a hippocampus. And then when my colleague was describing this New Zealand one, they found actually that their evolution has been convergent, and they're not actually that closely related, but that seahorse from the southern Indian Ocean wasn't a seahorse after all, it was a pipe horse. So even the taxonomic experts get it wrong sometimes.
Great, I have 2 questions. So how many eggs do they basically have at any given point in time?
So the Denise's pygmies that I was studying, between 6 and a dozen. So they're small broods, but they're quite large when they're born.
And then how deep were Tom, Dick, Harry, and Josephine?
Those guys were at about 11 meters, so like 30 feet or so. The most shallow I saw them was 4.5 meters, which is like 10 feet. I'm doing this on the fly, I think I'm doing well. But also they found them in submarines down to 120 meters. So yeah, maybe there's a much bigger population at depth than we know too. I actually couldn't hear you, sorry.
You talk about how much time you watch these, and how does your day look time-wise underwater? Are you up and down? Are you just doing it every morning, every night?
It was over sort of 6 months, different groups, and I would record in half-hour increments of, so I would start recording data of who was interacting with who. The data set was huge, and then I would do that for half an hour, and I recorded 100 and something hours like that. But when you arrive, you have to find them to start with. But I was just like hovering there with my camera and the clipboard, and reef folk, you can uniquely relate to that, I feel like.
That was amazing. Wow, it's awesome. And I think all of us can, as Richard said, relate to the finding and the looking for the next thing. And we really appreciate you making the big flight across the pond. And I'm going to keep all my fingers crossed that you find the dwarf tomorrow and the next day, so that it was really worth it to come. So thank you very much. And I know several of you were waiting to get your book signed. So we'll kind of wheel the table back out. And for those of you who are in line, if you weren't yet, just queue back up and you'll sign away. And we've got a couple more copies, I think, right? If you didn't get your book yet, and now you're inspired to get it. So for those of you coming tonight for The Love of the Sea, we will see you within about an hour. And for those of you who are not, thank you again for joining us this year, and have a really great evening. Be safe out there on the roads. All right, so